All right, everyone. So this is going to be a quick review of some of the concepts from the week 1 case study. As with all of our case studies, you should be referencing the Appendix 1 of EHDC in order to examine laboratory values and compare them to any lab values that you are provided in our case study. This case study will not provide you, this review will not provide you the answers directly. However, I will talk about some of the important things. So this first one's going to be very quick. Again, make sure for case studies you're always reviewing the EHDC Appendix 1 for anything to do with lab values. All right, so there's really two things we're going to talk about here. So for the urinalysis, what does protein in the urine mean, right? So obviously that means protein is not being retained by the kidney. There is lots of different proteins in the blood. Some of the most common are albumin and globulins. Globulins are a whole family of proteins, and albumin is a big transporter protein. It's super important for moving items through the blood, especially anything that is hydrophobic. It needs a transport protein, and albumin is one of the big examples of that. So really, when we think about the molecules that the kidney should be allowing through or has the ability to allow through, we really want to think about what those are and what they have in common. So obviously, water is one of those. Water is H2O, so 2 hydrogens and an oxygen, relatively small. It's 3 total atoms, right? We have electrolytes or salts, and those are going to be really single atom complexes. You have potassium, chloride, magnesium. All of these different electrolytes have the ability to either be retained or allowed to pass out of the kidney. So again, singular atoms. So the trend here is that items that should go through the kidney are small. Even other substances like creatinine. Creatinine is a pretty small molecule. We eventually hit a certain size of molecules, though, where the kidney pretty much should always be filtering those. Proteins are a great example of that. So generally, if proteins are able to leave the kidney via the urine, there is definitely something going on with the kidney, and it's going to be a structural problem. You're going to see damage with the glomeruli and the basement membrane because it's essentially something that is too large to normally fit through the filtering apparatus of the kidney. So our next other area to look at comes back to our patient chart. Again, review Appendix 1 of the EHDC. There's a couple things we could talk about here, but one of the things that I tend to see the most with this is everyone gets the white blood cells and the presence of nitrites. Nitrites are a bacterial-specific waste product. So if we have white blood cells telling us that there is some pathogen present, and nitrites tell us that that pathogen is more likely than not to be bacterial. And we'll talk about this when we cover the urinary system, but it's most likely to be something like a E. coli, one of the most common causes of urinary tract infections. Now, when we see these two things present, it's going to alter some other aspects of the urine. So when we look at our list of things, it's important to think about specific gravity. When we use specific gravity, we're really looking at the relative density of a substance, and that relative density is in the case of specific gravity, is going to be the density of the substance measured divided by the density of a reference substance. And in the body, that reference substance is water. We're always comparing to water. So if we have a specific gravity of 1.000, that is water, okay? Generally, we're going to see specific gravities between roughly 1.005 and 1.030. Anything below that is pretty abnormal and indicates the kidney is potentially having problems concentrating urine. And anything that's higher than that indicates dehydration, but can also be related to other things like medication effects, certain diseases, things like that. We want to fall right into that mid-range. Now, if we start to put stuff into urine that is not water, specific gravity would start to what? It would increase because the reference substance is the top of that equation, whereas the reference substance is the water, and that's what we are comparing the urine and its contents to. Okay, so keep that in mind. Specific gravity can be a very easy and insightful measure into anybody's health at any given time, and it's very cheap and very easy to track. So that's really the two things that I like to bring up with this first case study, because they're the areas that it seems that the students struggle the most with. Just again, make sure you're referring back to your anatomy and physiology notes and your Appendix 1 of the EHDC as a reference, because it will be very important to use that.